Hello, welcome to my channel Deep Learning Biology. My name is Dr. Vikas Mahardwaj, and within this video we are going to discuss making or breaking of carbon-carbon bond. Okay, in the previous lecture I have told you that within the cell, okay, you are going to see 5 major type of biochemical reaction, and in this video we are going to discuss the first one, that is making or breaking of carbon-carbon bond. Now here you must be knowing covalent bond. Covalent bond, it is being formed by sharing of electrons. So here if you see in between carbon and carbon, okay, you can have single covalent bond, you can have double covalent bond, you can have triple covalent bond. Now here if you see this bond between carbon and carbon, okay, it can break, or you can say there can be cleavage of the carbon-carbon bond. It can happen in two ways. One is homolytic cleavage and another is heterolytic cleavage. So here we are going to discuss that carbon-carbon bond, okay, it can break, okay, either by homolytic cleavage or heterolytic cleavage, okay. Now here if you see, let's suppose you are going to have this situation that this is carbon, okay, it is bonded with hydrogen, okay. Now here let's suppose this carbon is being sharing electron with hydrogen. So here there is sharing of electron between carbon and hydrogen, okay. Now if the situation is homolytic cleavage, in the homolytic cleavage what is going to happen, this shared pair of electron, it will be equally distributed among carbon and hydrogen. So here what you are going to see, you are going to see formation of this one, okay. So this is known as carbon radical, okay, and what you are going to see, the formation of this hydrogen, this, okay. So this will also have single electron. Now this will be known as hydrogen atom. So here if you see this bond, okay, it is being cleaved in a homolytic manner. What you are going to see, you are going to see formation of carbon radical and hydrogen atom. Both are having the 1 electron, okay. This shared electron has been distributed equally among the carbon atom and the hydrogen atom. So this type of cleavage is known as homolytic cleavage. Now here within the cell, in between biomolecules, you have bonding like this way also. Let's suppose it is carbon-carbon, okay. Now let's suppose here you are going to have sharing of electron, okay, and now let us suppose there is homolytic cleavage, okay. We are discussing about homolytic cleavage. In this situation what you are going to see, you are going to see the formation of carbon radical, okay, and another carbon radical, okay. So here you can see this shared electron has been distributed equally, okay. One goes to this, another goes to this. So here both these are known as carbon radicals, okay. So these are very reactive. We will discuss more about this. So here you can see in the case when we are going to have homolytic cleavage, okay, in one situation what we have seen, there is formation of carbon radical and the hydrogen atom, and here you can see there is formation of carbon radicals, okay. Now here let us discuss what is going to happen when we are going to have heterolytic cleavage. Okay, now in the case when you are going to have heterolytic cleavage, keep in mind one atom keep the shared electron. Okay, so in heterolytic cleavage one atom keep the shared electron. So here let us discuss the situation. Okay, this is carbon, then this is hydrogen. Okay, now let's suppose there is cleavage here, and let's suppose this is now heterolytic cleavage. In this situation what you are going to have, okay, let's suppose at this moment this carbon is going to retain the shared electron, and hydrogen is not going to have the electron. It has donated the electron to this carbon, so now it is going to have positive charge. So here you can see there is formation of carbon. Okay, so this is going to carry negative charge. Okay, and this will be known as carbanion, and this will be known as proton. Okay, so here when you are going to have heterolytic cleavage, what you are going to see? You are going to see the formation of carbanion and proton. Okay, now let us take another situation. Okay, so in another situation, let's suppose this cleavage occur. Okay, and what we are going to see that the hydrogen is going to retain the shared electron. Okay, so here what you are going to have, carbon is going to have the positive charge. Okay, and let's suppose this shared electrons they goes to hydrogen. Okay, so in this situation what is going to happen, hydrogen will carry the negative charge. Okay, now try to see here, this will be known as carbocation, and this will be known as hydride. Okay, so hopefully you are going to remember what is carbanion, what is carbocation, what is proton, and what is hydride. Now here you can also see the situation when we are going to have this heterolytic cleavage, okay, in between carbon. In that situation what you are going to see, if it is heterolytic cleavage, let's suppose this carbon is going to retain both of the shared electron. In that situation it is going to carry the negative charge. Okay, and let's suppose this carbon is going to have positive charge, so it is not going to have the shared electron. So this will be known as carbanion, and this is carbocation. Okay, so hopefully it is clear to you what is homolytic cleavage and what is heterolytic cleavage. Now I just want to give you 3 examples. Okay, you must remember. Okay, you must remember these examples. Okay, the first is example of reverse of aldol condensation reaction. Okay, hopefully in your chemistry class you must have read what is aldol condensation reaction. Please have a look. I just want to tell you, you must keep in mind, okay, which reaction is actually the reverse of aldol condensation reaction. So here if you remember the steps of glycolysis, in glycolysis what is going to happen, glucose is metabolized to glucose 6-phosphate, glucose 6-phosphate is metabolized to fructose 6-phosphate, and fructose 6-phosphate is metabolized to fructose 1,6-bisphosphate. Okay, now here the fructose 1,6-bisphosphate, it is going to have 6 carbon. Okay, it will be metabolized, okay, by the action of enzyme aldolase. Okay, yes, this reaction can occur in reverse direction also. So what you are going to see, you are going to see formation of glyceraldehyde okay, 3-phosphate plus dihydroxy acetone phosphate. Okay, now here if you see, this is also going to have 3 carbon, this is also going to have 3 carbon. So here what you are going to see, there is a cleavage. Okay, so this is actually the reverse of aldol condensation reaction. Okay, so here this 6 carbon fructose 1,6 bisphosphate, it is being lysed, and what you are going to see, the formation of glyceraldehyde 3-phosphate and dihydroxy acetone phosphate. Okay, so this you keep in mind that this reaction of glycolysis is example of reverse aldol condensation reaction. Now keep in mind another reaction, that is you must have read in your chemistry class, Claisen condensation reaction. Okay, now here I just want to tell you, we are going to discuss this reaction a lot. Okay, synthesis of citrate. Okay, hopefully you remember during the time of TCA cycle. Okay, what is going to happen, acetyl CoA, it is going to condense with oxaloacetate. Okay, so here it is going to have 2 carbon, this is going to have 4 carbon, and what we are going to see, synthesis of citrate. Okay, so this reaction is being carried out by citrate synthase. Okay, this is going to have 6 carbon. Okay, and here you are also going to see the release of this coenzyme A. Okay, so this reaction, okay, it is example of Claisen condensation reaction. Okay, yes, within the cell you are also going to have decarboxylation reaction. In case of decarboxylation reaction, what you are going to see, you are going to see a release of carbon dioxide. Okay, here I just want to tell you the reaction catalyzed by the enzyme acetoacetate decarboxylase. Okay, and this enzyme actually lead to the formation of ketone bodies. Okay, and that we will discuss in detail when we are going to discuss how ketone bodies are being formed. So this reaction, in this reaction what we are going to see, we are going to see release of carbon dioxide. So here we have discussed how breaking of carbon-carbon bonds, okay, can lead to various reaction within the cell. Yes, there can be making or breaking, there can be making or cleavage of carbon-carbon bonds, and we have discussed clearly what is homolytic cleavage and heterolytic cleavage. Hopefully you are going to like the video. Please do subscribe and share this video also. This is also important. Please do subscribe to my channel Deep Learning Biology. Thank you.